suggesting trying to pick up a rock in material like
15
that.
meters away.
Yes.
I wonder, Pilot, I wonder if I can see a black rock off at about 9 o'clock on the seafloor directly ahead of us. In fact, there's several black rocks, and we are up in the white material. As long as we do not go above 250, and I think those are below us. Those might be good targets for collection.
Yeah, you can follow me.
Copy that.
I think you're good. Just make sure the ship doesn't go any farther northeast.
Copy that.
That would be bad.
Yeah.
There's more sulfur there.
Good question. Good question.
So we're talking about this rock, Watchley?
Yeah, it sounded like that was one she was describing. Science, you listening to Pilot? He was asking, is this the rock you requested?
Yeah, this is the rock I was thinking about. Let's take a look at it.
It's interesting how this kind of white sulfur-coated area and the yellow kind of fluffy iron-stained areas are so distinct.
Yeah, they really are. It's as if there's just one type of fluid being involved in one area and another in a different area.
Nav, could
Different area.
Nav, could
Bill, do you have any idea how that could
please
take place?
Down to 30.
You know, within a single vent
Video, you want to
area
zoom?
in a volcanic cone.
Here's a close-up of the rock.
Well, you're probably looking at
Thank you, Pilot.
competition for sulfide from a variety of compounds, and so it's just that balance, the amount of excess sulfur, the amount of excess iron. And we did, I guess on Northwest Rhoda, we do see that move from white to yellow matte, which is where Craig goes to collect. And it seems to be, even though it's all the same hydrothermal system, it's just on that edge of who's got the sulfur at any particular time, I think, and what the concentration is.
Okay. Bill, do you think that this rock looks like one that we should pick up?
Want to zoom out and get the whole rock video?
Bill, do you like this rock?
in situ?
Can you zoom out a little bit?
Looks like it's
Can you zoom out a little bit, Pilot?
Yes. Video, can you zoom in?
Yeah, thank you.
or out.
In.
In.
Request from shore.
Partial. I mean,
So, Bill, what's the verdict on this guy? It's got some fuzz on the side on the left, I think.
all around. Looks like it's attached. Pilot.
Might be. It is unusual enough that I'm not sure.
It's a kind of funny-looking rock.
Yeah.
I'm wondering if it's
Solid.
semi-consolidated ash, and that if the manip drops down on it and tries to squeeze, it's just going to crumble away.
That's what it looks like to me.
Looks like the right-hand side has been broke off of it.
Do you want to go hunting for a different sample?
I don't, I haven't, when in these areas with the map, there's not a lot of big rocks.
Yeah, that's right.
Why don't we try and sample it while we're here?
You do want to try to sample it?
Yes, please.
Okay. Pilot,
Yes.
we do want to try to sample it.
Okay.
Thank you.
Video, will you go wide?
Turning on your hydraulics for you.
Excellent.
Shore side, you just went static again.
if I'm imagining.
It's just going to crumble.
I heard that.
Bill, this is Deb. Where do you think you see the shimmering so we can all look?
Well, when we were zoomed in on that rock.
Okay, do we have
off to the
hydraulics?
side, it
Yes.
seemed
You
like,
take Mini Zeus,
I
put
have.
it up on your screen.
There were
Mini
kind of
Zeus,
some
and I'm taking
waviness to the video that kind
Here.
of looked like shimmering, but I wasn't 100% convinced.
Okay, cool.
Go ahead.
Let's talk about potential flow on the right-hand side of this rock.
Sorry, can you say again? Okay, but you want me to try still? Okay. Thank you.
Well, we'll find out when you kind of nudge it.
Yeah, first we nudge. Because we don't want to put a lot of stress on the manipulator.
Do you want any zoom?
No, that's alright.
Ooh,
So it is loose.
looks kind of solid.
Cool. Yeah, it is loose.
And heavier sediment. Notice it didn't come up in the air or water.
Yeah, I think the sediment is, as our videographer said, heavier sediment. So probably larger grain size.
Okay, so first, want to do port slider in.
Copy that.
Yeah, port side with the lid.
We're gonna end up putting it in the port rock box.
Yeah,
You want to put it in the rock box or the bio box?
Rock box.
Rock box, okay.
it's on the starboard backpack. Not to be confused.
Yeah, it does.
Oh, do you want to
Yeah,
put it in
starboard
a
wing
bio
and
box?
port.
That's fine with me.
Starboard wing or port?
It's too big for a bio box.
It's too big for
Yeah.
a bio box. Yeah, I think we already have stuff
So
in the bio.
port,
Yeah,
port wing
Port
and
wing.
board.
Port wing and okay.
That's
copy
fine.
Yeah. And tilt the mini Zeus down a little.
that. Copy
we
that.
can put it in a bucket of water as soon as we.
Okay. Yep.
There are copepods on that rock, so I don't know whether you have any any ability just to look after the fact and look at the surface if there's any of the little copepods left.
Verena, what we'll do is when we bring that up, I'll go ahead and pull off some of that mat and put it in some methanol. And I'll also try and put some in for DNA. The DNA, of course, is going to have the bacteria in it too. But
Yeah, the Smithsonian has a wonderful collection of these kinds of copepods, if they are what I think they are.
Okay.
So morphological would be great if you have it in.
and ethanol. Okay, sounds good. We'll do that.
Yep, ethanol's great. Yeah.
What's our deep? 260.
260, according to the ROV.
260. Thank you. I know.
Beautiful.
So, Watchley, do you have a
Very
spec on
nice.
that, a name for that sample?
Was that nav?
Okay, want to move
Yeah.
the mini Zeus over for me?
I just put
Maybe,
rock
yeah.
with bacterial
Copy.
filaments.
Great.
That's as fast as I can go with it. Sorry.
No, that's okay.
And
All right.
Pilot, was that port inner?
Oh,
No, that was, you have a little chart there. That was
That's-
port forward
Port forward.
PF.
okay. Moving Zeus, mini Zeus back and disabling hydraulics.
Collect.
Before we lift off, do you want to look into the sediment of where you pulled the rock?
Sure. Right here.
Yep, maybe our geologist.
Oh, you know what? Video, I forgot to rotate it for you.
I noticed.
Are you going to forgive me?
I'll forgive you. Put it away pretty quick.
You can remind me next time. You'll let me.
Wait.
Say again, watch lead.
Say it again. You're muted.
Will do. We forgive you. We will remind you next time.
Okay, thank you. I expected it to crumble, actually, so when it didn't,
You were
just put it
so
away.
excited?
Flush with success.
Yes,
Yes,
that's the word.
indeed.
As a reward, he's
ready to
going
do
to
the-
zoom into the sediment so that you
Right.
can see how fine the sediment is.
Right. He was ready to do his victory dance
Okay, video.
to 80s music. Oh, what is that? What is that? What is that orange stuff?
Sorry.
Video defines it as flocking stuff.
Yeah, I know.
I think so.
know.
Little snail with its proboscis thing out at the bottom right, or whatever you guys call that thing. And tons of those little microscopic flea thingies.
It's really interesting. The snail seems to cover itself with the ash. Great camouflage.
Yeah, we had ones before where in an area that was very more black and white, and the snails matched that bottom too.
Comes one of your
a lot
worms
more
on the
white
right.
than here.
Oh,
and
yeah.
There it goes.
Zooming along. It does sort of look like it's poking around for something to eat. Didn't like that.
And that is the crater. So we have a little bit farther to go uphill.
Very
and then
little.
we'll basically be at the, don't know what you would call it, the end of this little canyon. And there'll
Yep.
be nowhere to go except back down.
Well, we can go laterally.
Little puppy jet crawling around.
We can go laterally along the contour, just staying below 250 to starboard.
Does that work for you, Bill?
Sure, yeah. Let's start heading toward.
This is
Do we need one more ship move?
how he's 15 from the wall there and it's angled in. Yeah, maybe. Oh, and he's at 300, I see. Yeah, maybe just like 10 meters at 300.
Interesting square block inside of that orange.
Yeah. Is this the rock we should have gotten?
I think so.
Bridge nav. Yeah, we'd like to move 10 meters, 10 meters,
Bill, this
bearing
is
300,
almost exactly like the setting on
0.2
Pacific
knots,
2,
please.
where we found the flatfish in the map with the same snail.
Huh.
I was wondering if we might encounter it here.
Correct, thank you.
No. there's a You'll be diving there in a couple of days.
Well, while I
Really
wait,
looking
I'll
forward
look
to
a little
seeing
bit
whether or not
starboard here.
you find it there.
Yep.
Give you
Because
just
it
a little
occurs in
bit.
the hundreds per square meter.
Wow. Okay, we'll be on the lookout.
And it's the same snail as there?
I'm almost positive it's the same snail that we encountered. We called them dumplings at the time. We couldn't figure out what they were.
Right, because they're all coated with stuff on their shells, right?
Yeah.
Things
And these are doing the same thing,
possible
but the coloring's a bit
crab,
different. But the,
right
the
edge of
foot
screen.
and the siphon and the shell
Sticking out there?
shape
Yeah,
are all
probably
It's a little
spot
spot
lobster.
lobster, yeah.
Interesting.
Verena, what are some of the characteristics that you would need to be able to describe this down to the species level?
Want to do a zoom there, video? back
Snails in the hand, I'm afraid.
Yeah.
Oh, so actually, I mean, I'm not a describer of anything, but to be able to look at a description and then match it with the animal in the hand, you're looking at shell characteristics, and you're looking for snails, usually just the way the shell itself is formed, but there's also characters of the way the head is, and some internal anatomy, but most people working on snails just work on the shells.
Yeah.
So this species was described by
reacts to that thing falling on top of him.
some Japanese systematists who collected it up in the Izubonan Arc.
Thank you.
Well, we're going to leave the next watch in an interesting predicament.
Something white just left of that angular rock.
Want to go in?
Yep.
It's mat growing on a...
There used to be something. But it's not anymore.
Yeah.
Yep, you can see some more of that. The fuzzy filaments, bacterial filaments on the edge of the rock, actually probably all over the rock. Hopefully we'll be able to take a small bit of that off of the rock sample that we just collected, preserve some of it for later analysis. As we mentioned before, these samples will be publicly available. They will be, the biological samples will be archived at the Smithsonian Institution. The molecular samples, we're taking a small bit of each of the animals and putting those small pieces into a special buffer for DNA and other molecular analyses. They'll go to the Ocean Genome Legacy at Northeastern.
Look at that crack or line.
Sure. I'm
Say
going to
again,
stay
Pilot?
a little back, make sure it's not a fishing line.
Just commenting that it looks like
Yeah.
potential crack.
Do you want to zoom in
Sure,
on it there?
let's go up and take a look at that.
Okay. You can go
Please.
in video.
And then the geological samples are going to go to Oregon State University. But again, they'll be available for
It looks like a trail,
anyone
maybe.
who's
Yeah,
interested in
it's
studying
closer
them.
to
Put in a
no.
request
Yeah,
for
I
them.
was just staying back in case it was a fishing
I
line,
think
but
it's
it's
a
not.
crack.
Yeah.
I think it's a discontinuity
crack.
in the rock.
It's really hard to tell from this angle.
Well, I can drive up to
No,
it
I
now.
think it's an animal trail.
Yeah, I guess it's an animal trail.
Seen a
something
few
that-
of them.
Something that went
Wow,
along
it's
and
pretty
just
long.
ate the
A
mat as
couple
it
of
went.
places.
It's pretty long.
Very straight. He or
Pretty
she
straight, too,
knew where it
actually.
went.
No. But no, it's got meanders at the other end.
Yeah, what is at the other end?
Watch it be that
Well, maybe we can
flat
find it.
fish. Let's look at the end of the trail. Yeah.
I'm on my way.
Was it going uphill or downhill, though?
It actually is going off the edge of the screen.
Uh-huh.
Our ship moves
Yeah,
about
it's
done.
going very good. I'm at 256. Probably call that the end.
Mm.
Okay, video, you want to zoom in on it?
Yes.
Just rotating around. Go.
Looks like a Holothurian trail if I were to do
You think
mine.
so? Yeah, I don't
I'm
know.
an expert hunter of land-based critters. And I've been tracking these critters for 6 years on the ship, so I
All
will
right,
leave it to
so-
the people that have been studying them for 40 years.
All right, so Roland, our videographer, is calling this a Holothurian track. Those of you who've been studying these, what do you say?
There looks like there's something above lasers. Looks like there's some slight relief from it, but it's also a straight line, just upper right of center.
Okay, we can't do too much more upper.
Now, when does this shift end?
At
Isn't it?
2:30.
Oh, so the last watch is only 2
That's-
hours?
that's-
Dad, is it possible to get the quad screen
that's,
back on stream 3?
UTC. That's 2:30. That's 2:30
Roland's
a.m.
taking care
It's
of
an
that
hour
right
from
now.
Sure.
Because we're plus 11.
But yeah, it's
Thank
1:30
you.
now. But do
You're
they only have a
welcome.
2-hour shift on the last watch, or 3-hour?
Yeah, it should have been 1:30 then, right?
We could go look at the board. I thought it was thinking it was maybe been 2 as well. I'm not sure.
Oh, okay. Well, it's okay. We're doing well. I just thought they were coming in. Okay. I'm good.
I'm fine.
All right. So watch leads. I think this slope extends a bit ahead of us, but we're at 255 meters depth. So it's about time to turn to starboard and look at the
Okay.
cliff.
So we're definitely.
Agreed. Yes, sir.
So co-pilot, should we ask Casey to find an alternate co-pilot for a few minutes?
I'll come up and do that. No, just kidding. They laughed. They laughed at me. They were looking for an alternate co-pilot to come in. I volunteered my services.
We just ask Casey to find somebody. Oh, here comes Josh.
Welcome, co-pilot.
Well, we have an outcrop over
Howdy.
there on the right-hand
So
side.
we there's a cliff here. We came
Yeah.
up along it, and we're at our shallowest depth. So we're supposed to work at this depth along it, which is going to be very slow.
We go into port
To
Starboard.
to starboard.
basically downhill.
The other port.
Okay.
So I guess we'll just get you lined up with it. I'll just sidle along and look at it. I think you're probably closer than you would think. We've been
No,
keeping an eye on it.
Yeah, I can see.
Yeah.
I'm only like 15 meters
away, yeah.
Yeah. But how tall is it? It can't be that tall.
Well, it's part of that.
Uh,
We haven't been to the top. We followed this mat up, and they didn't want to look at the cliff until we.
Yeah.
Yeah, it's pretty murky up here. It's hard to tell
10, 30 minutes, 2 o'clock.
whether this exposure in the cliff face is similar to what we saw a little shallower, deeper.
Yeah.
And whether or not it has columnar joints too.
Okay, I think you'll have to tilt down and follow me around. I think we'll go until you're perpendicular to the wall.
Okay, is our plan to go Explorer's Wall now, I guess?
Yeah.
But
Copy that.
boy, that
We'll follow you.
white,
It's amazing, the pattern.
if it's a bacterial mat, it goes right up to the cliff face. And even up the cliff a little bit.
See that rougher texture in the white?
What's...
And then there's brown staining areas. Just,
Yeah, I'm looking at that orange
yeah, the orangey
What is-
brown.
right up at the...
Oh, the creature. Of course you're looking at the creature. What am I thinking? Of
What
course
is
you are.
that? What is that?
You want to snap to it?
I'm going up to it. It's definitely something.
I believe it to be 2 starfish.
Oh,
Or
that's
one.
why it's unusual.
Oh,
Or
could
one
be debris.
multi-legged
Oh, I don't think
starfish.
so.
Pile of sea
I
stars.
think it's a critter.
I think I agree with Roland. 2 stars.
I think it's got a lot of arms.
1 fish, 2
Okay,
fish,
you want to zoom in?
1
Geez, I wonder if this is one of those
multi-legged
weird sea stars.
starfish. Yes, it is.
The nubbly
Yeah,
ones
yeah,
like we saw
yeah.
before.
This is.
Coronaster, right?
Oh, wow. I love
You
these
got it,
things.
Shirley.
I'm learning.
I'm getting some
Chris Ma would be so proud of me.
fluid flow or temperature change.
Now he's mad now because you scooped him.
Take a look. Seems pretty gradual right now. I'm seeing 17.3.
Having difficulty focusing. Look like we're getting some shimmering in the water, whether it be temperature or current.
So these are multi-legged sea stars, and there's little bumps on the
We got the lasers on.
surface
Yes,
are
I can do that.
pedicelaria. I think they use those for defense, maybe.
Lasers are on.
See, I
Great.
didn't learn
I
everything.
think that would work.
I think Chris Maz still has a place in this because I didn't quite remember everything that he said, but I think they are for defense. But the other thing is that they can shed an arm, so if some predator comes and tries to bite at it, it can drop one of those arms.
Now,
It has a lot of them to spare.
Now, if it does that, I'm curious. Does that arm have any nerve action in it that makes it wiggle at all?
You see the
Like
crack
a
in the
you mean
rock
like
in
a
the top
lure?
corner?
like a gecko drops its tail and the tail goes flitting around?
I have no idea.
Yeah.
That would
Painting
work.
on the right-hand
I
side.
have absolutely no
may
idea. Verena is seeing
be a
some
flow
shimmering in the
source.
water. Has anybody
Where?
else seen that?
Right-hand side.
Roland, can you see shimmering in those cracks?
I am seeing. I'm not seeing it necessarily from the cracks, but when I image that starfish, sea star, it was difficult to get focus because the water was shimmering around it, and I couldn't tell if it was just the water near it or if it's something coming from this area. So we're investigating this crack on the upper right, and now camera right, to see if it's coming from there.
Yeah, temperature's 24°C, which is not unusual for this depth.
But we'll take a close look into that crack and see if anybody on shore can tell us whether they can see any shimmering.
I don't see any shimmering coming from it, and I don't see any
No, I don't.
No, I don't either.
hanging out, except
Okay.
for those
Nope.
More of those.
tube tracks on the wall.
Let's keep looking around. Can you go wide video?
I think
So
that's more
Shirley,
like fecal. Yeah.
just in my experience with working in this depth range, 24 is actually pretty high for 250 meters. Usually that's down around,
Can't back up
it can be
too
anywhere
quickly.
between 15 to 21.
Come back. No, I said I backed up.
Okay. Well,
Yeah, I wouldn't
Are
be
you guys seeing
surprised if this
24
is a
on the CTDs
hydrothermally
over there?
active
Okay.
area and
Are you seeing
and
24
very
on the CTD,
diffuse.
temperature-wise?
As we see so much bacterial matting spread out over the seafloor
no,
all
these things
over this
17.
area that
Okay.
we've been traversing.
Yeah. Okay, yeah, I guess they saw something we didn't.
Chris Ma here. If you have any questions about corn, I'd be happy to take them.
I knew you'd be listening, Chris.
Watch leader. Watch lead.
Well,
Yeah.
all of this interesting geobiology with the bacterial mats and whatnot.
That white thing at the
So
base of the
yeah,
rock.
you were correct.
Upper right
This is
lasers.
coronaster, and you
all
beautifully
that,
mixed
so
all of the
just there you
spawnings
can
on the
zoom in.
surface, which
It's
are
like a little piece of
carrying
rock, maybe.
tufts or pom-poms
Top of the screen
filled
now?
with these
Yeah.
pedicelaria.
Yeah, I think it's just a chunk of
This
rock.
is actually a very distant relative of, like
Rock
if you're
beams?
from the west coast, sunflower stars, the Pycnopodia,
something
and
under
if
the
you're on
ledge
the
at the
east
top.
coast, things like Asteria. So they've just taken that aspect of it and ramped it up
Right,
about
just
10
starting to
times.
wrap around.
You
It's
see
thought
that
that they're predatory,
circular object right off screen
but
top
we've
right?
never actually seen them use the pedicelaria either
I'll have to back up a
defensively
bit
or
to see what's on my right.
they're very similar to Brisingid in structure and overall appearance. So it's thought
coming towards
use
center.
them to feed that corn aster could use them as well, but they vary in size. Some of them are quite large. I mean, almost a foot to a foot and a half across, whereas that one was probably not more than about a foot across. But from what I'm told, they do feed on other
So you can start
Kynoderms,
following me around.
in
Copilot?
some places.
Copy.
Not sure exactly where.
Squared up to the
So
wall.
when the
Yep,
arms
as soon
are
as
dropped
I
during
pull up slightly here.
stressful moments, most echinoderms have the ability for the arms to sort of move off on their own for quite a long time. This particular species, I've not seen the full gamut of the time it can live without its arm. But I do know that there are some sea stars that the arms come off, and you can literally keep them alive on the arms alive on a water table for months. And
Wow.
you can give them little clams, and they move little clams from one end of the arm to where they think the mouth is, and then just
Oh,
pop it
gee.
off because the mouth's up there.
Oh, that's
So
amazing.
Things can get kind of creepy.
Oh,
So, Chris, is there a
That's better. So you're pretty squared up there.
How that functions.
Yeah,
in the
I'm
wild.
facing it directly.
Anyway, so
Excellent. I feel more comfortable this
looks
way.
like you guys are moving
Copy
on into away from the bacterial mess. So thank you for catching one of those on video.
It looks like
Makes my entire day worthwhile.
to the
So I have- wait, I have
there's
two more
there's
questions.
a jog in it.
don't go away.
Don't go away.
Yeah,
so,
there should be.
I see
We're
that.
guessing north.
That's right.
what's
Okay, we saw
I
it
know
before
there's
it
a
went away.
scientific term for when they drop off their arms, but I just can't remember what it's called, like excision or something like that.
For just
Autotomy?
Haha.
Yes,
autotomy.
autotomy. Okay, thanks. And boy, I had to reach back for that.
Vary among groups.
Okay.
The ability of which a sea star can autotomize its arm.
And what is there ever a piece of the disc that's on the arm?
In this group, the arms, when they come off, they're off.
What'd you find?
It's not like there are some families like the
Lighting's
Ophidiasterid
bad now.
where if you have a little bit of the arm,
We're awfully close to that wall.
I mean, if you have the arm and a
Yeah,
little bit of the
no.
disc, it
Swing
can
on.
regrow a whole animal.
Or you're pretty close too.
Something that's been
I'm holding
shown
pretty
in,
stationary though.
textbooks
I bet
as
if
a
I came
comet.
up, you would see that it's right in your face.
In my
too.
experience, most of the time when you see something like this,
But you're still 15
they-
and steady. So
the
keep a close
arms
eye on
don't
it.
regrow a whole body.
Definitely won't move any. towards
There
it.
are some
As long as the
sea
ship
stars
stays.
that can do that. And,
Oh, yeah,
and
I
some
gotta
in
watch
this
that.
group.
Okay, video.
But
Want to
it
zoom in
tends
this white?
to be more of the exception than the rule. So regeneration is possible. The disc can regrow arms, and I've seen some of these cornaster sea stars with variable numbers of arms lost to predators and such. It just does depend on sort of at what point you've caught the animal.
I see shimmering here.
Regrowing the arms at
Wonder if
different
that's not
There's
current.
lengths.
I think they're
So,
shimmering
they can
everywhere
sometimes
from
be very
just all the
small,
currents moving around.
and sometimes they can be quite, half grown or what have you.
Great. Thanks so much, Chris.
Yeah, you tend to find the really
Oh, wow, the
elaborate
surface current's 1.3 knots.
in
Straight
other
ahead.
sort
They
of more
pick up.
tropical.
Great. Thank
Yeah,
you.
video, will you go wide?
Yep, we're seeing shimmering water now.
Have a good night.
Yeah, you too,
Well, you
Chris.
can probably check that ADCP
Like a
from this
strong
depth.
current maybe pushing by, and that's what we're seeing with that
Yeah,
shimmering.
yeah. I was wondering if it could be prop wash too.
No, I don't think
No,
so.
I don't think so.
Actually, we're seeing temperatures on the CTD of between 16.5 and 17, a little less than 18 C. I haven't seen anything in the
The
20s.
outcrop wall that we're looking at looks very massive. I don't see columnar joints the way we did lower down.
What?
And I'm wondering if there's
No, 1 degree.
some sort of hydrothermal overgrowth that's kind of smoothing out the surface.
Yeah, oh, yeah. You're looking at bottles, yeah.
So I have a question. Is it possible to have a cold seep?
It is definitely possible to have a cold
I mean,
seep.
in this area, like where there's
Oh,
volcanic
in this
volcanic,
area?
yeah, in an area where there's that kind of volcanic activity.
I think if there had been an eruption as short time ago as 2014, that it would still be warm. Cooler than super hot, but not cold. It's so massive
I've seen
here.
it come
though.
down a little bit.
Wow.
I mean,
Yeah.
2 meters.
Yeah, you can come down a little.
Okay, I can see you might be going down
Maybe-
here shortly.
Yeah. Maybe we just want to have you back up like 5 meters.
Sure.
That. So right now you're looking straight down at me, right?
I am.
90 degrees.
Yep.
Yeah, that might make everything more comfortable.
Okay, let's
I
let's
see.
do that.
Watch lead, nav.
Go ahead now.
Yeah, the CTD's temperatures are much lower than you were thinking. The temperature you were seeing was internal to the electronics housing on the ROV.
Oh, thank you. Thank you.
Yeah, so we're seeing temperatures
So
in
you
the
wanna move
16 and a
about
half
5
to
meters back?
17 and a half
So, yeah.
degrees C
Copy
range.
that.
Okay.
It is interesting.
That's pretty
We're
normal.
seeing a difference between D2 and Sirius of over a degree right now.
Ah,
With-
okay.
With Sirius actually warmer
Well, that makes sense.
because it's higher up.
Nav, co-pilot, could we get a ship move just back about 5 meters, just so that we back off this wall just
Tell
slightly?
me what.
Oh,
back is.
we're looking at 186, 187, around there.
Bridge nav. Could I get a move, please? 5 meters, bearing 186, 0.2 knots.
We're trapped up here then. We're in a dead-end box canyon here. You want to zoom on this?
That's correct. Thank you.
Zooming on the bio.
We'll be asking you for half a meter soon.
Oh, look at filaments.
Filaments, yep. White ones and yellow ones.
So everyone keep
Max
a close eye and make sure the ship doesn't go north.
Copy that.
There's a brittle star, I think, in about
Cool.
Yes, there is.
4 o'clock
Yep,
from the
there
center.
Sure is.
Very, very tiny.
Do you have any more zoom video?
No, that's maximum.
Okay. So watch leads. This is pilot.
Yep, go ahead.
We're kind of just sidling down this wall. We've already come up it and glanced at it a few times. Is this the goal for the next 2 hours? Just kind of work along the base?
Yes, that's correct.
Okay. Who?
Oh, our relief is here. Okay, the ship's moved about 5 meters.
All right. Yeah, Sirius
What do you
is
think?
looking a
Should
little better.
we go take a look?
We should just continue on this way.
Oh, okay.
Hey, pilot, can I follow you around too?
No,
you
we'll
wanna just
keep
keep
you
facing this? Okay.
facing it, and then we'll move you kind of sideways. We can actually, I guess, follow me like 15 degrees. We'll kind of split the difference.
Okay, copy that.
There's-
Look, your laser's about to touch an urchin on the wall. To the right.
Watch lead, pilot.
Go ahead,
The
pilot.
other dimension I didn't ask about, do you want to stay at about 260 meters or kind of follow the base and, go down to 270, 280?
That's a good question. Let me ask Bill
Okay.
Chadwick. Bill Chadwick, sir, our pilot would like to know if we should stay at about this depth or whether you would like to take a look at the base of this outcrop as we go around to starboard.
I think you're free to change depth.
Afternoon,
My
pilot.
only suggestion would
Afternoon.
be just continuing to traverse.
We just
the east
backed you up a bit,
see
and I
if
came
there's,
down. So if you want to come down a few meters,
Sure. Ooh,
outcrop
22 meters.
yeah, I've been coming
go in
down.
that direction.
Come back a little.
So,
First, we wanted to
whatever
back you up.
guys want to
Yes.
So
Okay,
right where we are, the
Thank
wall
you.
is about east-west.
so
Okay.
pilot,
Go ahead.
why don't we try to follow the contact between this ash layer with the white material on it and the base of the outcrop as
Okay.
we go
We can do
to
that.
starboard.
So, yeah, they just asked to follow this interface between the vertical wall and
Shirley,
the
do you
flat
think
area.
that's an urchin on the wall
I don't
of the
know the best
outcrop?
way. Right now, the wall's about
Kind of
east-west.
at 2 o'clock?
So we would basically
Yeah, I'm not
move
sure.
you-
o'clock?
Not sure.
It's a little round thing.
Yeah.
Don't know what it is.
We would basically
Video
move you
thinks it to be an urchin.
due east.
An urchin. I'm getting good.
Okay.
And I think that thing at the bottom, I
I would keep looking ahead of you, kinda-
Yeah, looking at the wall.
Yeah.
But I might angle slightly toward the-
Yeah, so he
Direction
angled
motion.
you like 50,
Huh.
or he's at 22.
Oh,
so due
another
north.
one?
Great.
And to the
Okay.
right of that, if you follow the
Um,
crack of those rocks, I
So,
see
but
about,
of course, it's going to start
4
curving that way, so you'll have to keep adjusting
urchins
yourself.
in
Stay,
this area.
keep serious
Really?
about. I did,
Where?
to the
15
right,
meters
to
is
the
fine.
right,
Yeah.
Yep.
potential
definitely not closer.
Ctenophore, is that what that thing was?
I don't know.
Tina
Maybe we
Ford?
can zoom in on
That's
that
about
when
it.
they get
Okay.
situated.
So real short hops.
Yeah, very careful. This
Pilots,
is
when you're ready, I wonder if we could kind of zoom in on that white thing right in the center of the screen.
we'll do watch lead. We're doing a change, and then I'll pass it on to Josh.
Got it. Thank you. We're changing out our front row folks. Our navigator, pilot, co-pilot are switching off.
The more I look along the base of that rock before it actually goes into the sediment, all along there I continue to see what looks like those urchins. I probably see 6 or 8 of them in the screen right at the before it climbs up the cliff.
Yeah, I see what you mean now. I think.
Yep, sure does look like that. Ctenophore.
Yeah, it does.
Oh, and now I see all the urchins.
Yeah, I do too.
All right, watch leads. Just had a shift change in the front row. We now have Dave Casagrande and nav, Josh Carlson as pilot, and I'm Dan Rogers as co-pilot. And of course, Roland Bryan, video engineer.
Let's come on in, Roland.
All working with the Global Foundation for Ocean Exploration.
Thanks very much.
And here we go again, another
Wow.
Ctenophore. This guy's not as white as the previous ones, And again, there are a lot of the shrimp, the same shrimp that we saw, and the cracks in the rocks.
I think it was Scott
This
France.
one has a lot more color in it.
Yeah, I think Scott France was saying that some of the ones he's been working with or has experience with were orange. Or maybe that was Brian. Or maybe it was both of them who said that.
Now, you said earlier that...
Oh, see, now you can see the combs along the... Yes, you can. You sure can.
So the little white hairs are the tentacles. No, those are the shrimp feelers.
Yeah, see the
I
orange
see.
stuff, yeah.
Hi guys, I'm on the phone this time. If I can offer any information.
And who is that who's speaking?
Hi, it's Scott France, University of Louisiana, Lafayette, calling back in.
Tell us about these, Scott.
So we're looking at a ctenophore. This is a benthic ctenophore. We've seen, for those who have tuned into the expedition for some time, we've seen lots of ctenophores in the water column. They look like little ovals, and they swim around using cilia. And usually in the light, you see those cilia when they beat, kind of like paddles of a boat. They reflect light back, so you see these rainbow shimmers
settled
over
out
the
right
body.
now, but we're gonna.
This is an interesting group of
keep a very
ctenophores
close
which is
eye
adapted to live on the
things.
substrate. They've really
We
changed
want to
their
pay
shape.
particular attention to the
And
sonar.
so instead of
We
being
don't want
a
Sirius to
small
get
oval that
much
can
closer
kind of
to the wall than we are now.
such that the mouth is now against the substrate, and they're kind of flattened, except you see those two large
15
lobes that are
meters
extending out
cliff,
from it.
yeah.
Okay.
Those are tentacle sheets. And so the ones that swim around the water column, many of them have these very long tentacles.
Very
this is
small
where the
moves,
tentacles would be housed.
small and precise
and they would
moves,
normally be strung
to
out
try to
much
follow
like
the
a fishing line. They have
wall contour
specialized structures on
without
those
putting the
tentacles
vehicles in danger.
that
Bearing
fire
of 180
basically sticky
is
globules so
still
they
away
can
from the
capture
wall.
their prey.
Yeah,
They
that's,
essentially
you know,
glue the prey
as we
to
make
the
these
tentacles,
moves, just
and then they'll withdraw the tentacles
always be
like a
aware of
fishing
what
line
direction we
and
might need to
move
go,
it to the mouth
pull off the
and,
wall,
feed off
get
of
us
it.
out of harm's way.
Video's
And
clear with you.
ctenophores are a group of their own, but I guess historically people have thought they're kind of closely related to the cnidarians, which is where the jellyfish are. But in fact, newer studies have scientists kind of scratching their heads. It's a little bit unclear
on the Sirius
what these things are related to. They seem to be much more advanced than was originally thought.
So maybe bailing out in that direction is not the best.
Thank you, Scott.
And Scott, what do you think?
Yeah.
So are
There's
they
some
using the
urchins along
tentacles
the
to
top.
feed and draw the food down, or because you mentioned before that they're
And below.
attached to the substrate or stuck to the substrate, and that's where they're feeding. So are they feeding off of what's on the substrate, or are
Your
they
mic
doing a
is really
combination
low.
of
How's your
capturing
positioning of
particles along with those tentacles as well?
So maybe a bailout more like 120.
They are feeding on the substrate. At least I have not read anything like that. Instead,
Yeah,
they
and I mean, we wouldn't want
place to perch
to get moving.
and instead use the tentacles like
Quickly.
a fishing line.
Okay.
Want to move fast.
And so that's
Yeah.
where they would get their food out of the water.
It's more like if
and
we
there's
start
a lot
swinging
in
into
the
the
water
cliff,
column,
you'll
as
want
we're
to
saying here. I mean,
do a
there's a
short
lot of
hop
stuff.
Okay,
backward in
Partial.
order to pull us off of the wall.
got it.
Your mic is
Wait.
closed.
Volcaniclastic rocks of various sizes, but they're all covered with a fine mesh of white bacterial mat, I guess, right,
Video's
Shirley?
clear with us.
And we're looking at, of course, an echinoderm in the center of the screen. Yeah, quite a few of those around. You'll see them also perched at the base of the outcrop wall.
Hey Nav, can you let Andy know that he can listen to Survey 1?
So for those of you who are
So we can get down
just
on a
joining
headset.
in, although I imagine that no one from the East Coast is just joining in at this time, but you
Yeah,
never
he's
know.
on that.
We are broadcasting
partial.
live from the NOAA ship Okeanos Explorer, which is America's
Bridge,
ship for
are we
ocean
now?
exploration. We're coming to you live from the
Can we change
Mariana
our
Trench.
bailout plan to bearing
We're
of
exploring some
120?
unknown areas. To the best of our knowledge, there's never been any ROV dive. This place where we are right
And
now which is the
we're
IEC mount.
just a note. We're in a canyon here, so we're probably not going to want
My
any
name is
large
Shirley
moves.
Pomponi. I'm the biology science lead on this expedition. I'm from Harbor Branch Oceanographic Institute,
That's perfect. Thank you.
Florida Atlantic University, and I also head up NOAA OER's Cooperative Institute for Ocean Exploration, Research, and Technology. And my partner in crime is
Patty Fryer. I am a research professor at the University of Hawaii, and I am here looking at a volcano that's 200 kilometers west of the Mariana Trench. The expedition that we're on is called the Marianas Expedition, and so we're looking at a whole bunch of different types of features on the seafloor in and around the Mariana Trench. Yesterday we actually dove in the trench on the inner slope of the trench wall, and I see a tiny little gastropod
Actually, it's a crab.
in the center of the screen.
Crab inside of it.
It is a gastropod shell with a hermit crab in it.
lots of things it can pick off of the bottom to eat here.
Boy, I'll say.
Scavenger delight.
You ready to pull up?
Yep, come on up.
Often at hot vents, the bacteria are the base of the food chain, and you get lots and lots of organisms.
A huge collection of those on the right, the urchins.
Yep, a lot of urchins now.
It's interesting that these urchins nestle
the
pretty
wall.
much along cracks or at the base of the outcrop, right where this this aggregated talus, the fine particles that have piled up
So
on
Josh,
the
I'm looking
seafloor
at
from probably from an eruption
when
at
we
the
do
summit
start
of
to
the
move,
seamount.
keep us kind of or parallel to the wall.
So, Shirley, why would you think
reading
that
about
they
50
would
degrees
like to
starboard.
hang out right at that crack?
So,
or, you know, right
of
where
our
the
current heading.
the loose material contacts that
Something
vertical
like a
wall.
080, and maybe we can do like a 10-meter
I
hop
have
or
no
something.
idea.
Sure.
I'm
Go
just
ahead,
wondering
partial.
if
Unless there's some, something that's accumulating there that they're eating.
Yeah, let's go
Where's
ahead
all-
and-
okay, so now it looks like it's
Partial.
this urchin
Yeah,
is all congested.
camouflage. It's camouflaged.
Yeah, let's go ahead and call in
One on the
It's
right.
like a
10
decorator
meters,
urchin.
080.
Do you think they pick up that kind of stuff to
Normal speed,
kind of
0.2
hide
knots.
themselves from predators, or
What is
Let's
that
just do
one?
this
0.1
just gets
for
stuck
now.
as it
And we can
falls
speed it
down
up
the
if we need to.
wall?
We're
Honestly,
not
don't
going anywhere
know.
too quick.
Now, what is
Oh,
on
yeah,
that
'cause that was,
urchin?
Scott was asking that before. What is the, because he saw some before, the disc. What?
Bridge ROV nav.
Oh, that, to me, that also
Can
looks
we get
like
a
some
move,
brown algal
10
debris.
meters, 080 bearing, and
shell.
0.1 knots.
Yeah, I don't know. You think?
Maybe from
I don't know.
It's a head
Copy.
scratcher.
Yeah, anyone on shore?
I'm getting tugged. Yeah, I'm moving in your direction.
We're really lucky the way this works is that Shirley and I both have specific areas of expertise, but we're able to communicate
at the, wall.
with scientists
Yep,
on
I heard
shore
you.
that
Okay.
can expand the scientific knowledge base that is looking at what you're seeing on the screen.
And thank goodness, because my expertise is in sponges, and we haven't. I don't think we've seen
Don't know
any
if you
today,
relay
have
D,
we?
but at 250
And my expertise is in
meters
rocks,
seems to
and
be-
all these rocks are
Yep.
covered with biological goo,
Thank you, Bridge.
Come on in, Roll.
which makes it very difficult to see the rock.
We are moving.
Yeah. So Tara Harmer Luke we were asking about. Patty was asking about why these urchins have all this material on them, and Tara commented that since so many of them have some kind of material associated with them, she wonders if it's some sort of camouflaging mechanism. Indeed, there could be some kind of just like they're decorator
Urchins.
urchins. Yeah.
Yeah, thank you, Tara. That's what I was wondering. It seems kind of like
Yes,
they're
it's
actually picking
fairly
it up.
common that urchins, especially
Right,
in
come
shallow
out.
water, they shade themselves from light. but even you will see them in deep water in the northeast Pacific, where they're northwest Pacific, where they're covered with rocks and bits of algae and anything that just that seems to be something that's partly protection from predators, but also in high current areas, they keep themselves anchored down with stones on top of them. So I think it's just something that urchins do in general.
Wow, that's amazing. That's fascinating. We're beginning to think.
Got a little more slack now.
Yeah, thank you.
I can come down a hair too.
Yeah.
Co-pilot, we're about halfway done with
All
that.
right, come on in.
Okay, thanks, Nav.
How massive these outcrops are.
Yeah, it's been pretty surprising to see how
All right, come on in, Ron.
smooth and, as you say, massive the wall of rock that we're looking at really is. A little bit lower down the slope, we saw columnar jointing in it, and I'm just wondering if this whole feature that we're looking at
really
is the face of a dike, a vertical intrusion of magma into the flank of the volcano we're sitting on, that cooled underground basically until subsequent eruptions just knocked the top of the volcano apart, and the more resistant to erosion material, like the solid, massive dike material, just is what's left over.
Yeah. Come on up.
And there could be some larger, intrusive bodies too, like stalks that are more like plugs, that are in the core of these Mariana seamounts.
Co-pilot, our move is just about done, but I am seeing a target in the direction we're moving, 22 meters.
Yep. See that as well. Yeah, we'll keep doing these 10-meter hops when he needs some tether.
They were sort of all exposed when there was a big landslide at that volcano too, so this kind of reminding me of that.
That sounds very likely.
These volcanoes grow by most of their eruptions are explosive, and so the material that comes out is all fragmented. So there's rocks and ash and that get thrown out, and they just pile up around the vent and form a big cone. But it's all loose. The more it piles up, the more it gets unstable, and periodically there are these big landslides that move all that loose material down the slope, and what's left behind is all these really massive, solid pieces of the interior of the volcano that don't stay in place. So,
did we turn off the trails on High Pack? For any reason in particular? Or they just really
weren't
erratic?
you on the Ring of Fire dives for NOAA where you first observed, absolutely the first time ever that people observed
you need to
an eruption on the seafloor was in one of these guys?
Yeah, that's right. In 2004, we were out here on an expedition that was funded
come down
by
a bit. Give me more tether. Feeling pretty safe now.
We encountered an eruption going on right in front of us. The seafloor about a depth of about 550 meters, and it was really jaw-dropping. First time anybody had seen a deep sea eruption, and it was luck, but it was also that that volcano was active for years and years, pretty much continuously. So I had the chance of encountering it.
And Bill, how did you observe that? Was there an ROV or some lander, underwater camera? How did you do that?
Yeah.